Hi Melissa, congratulations. So the paper is out at last in Science Advances. How do you feel?
Yeah, I was very excited about this. It's really great to finally have this piece of research out at last. And it's taken a long time to get it together, and a lot of us have worked really hard to form this piece of work and understand how bacteria look, what the outer surface is like, and how that changes to a more elegant structure from these textbook definitions that make it sound so simple.
Well, I think that is very fundamental to science, and you've really used a wide range of expertise and methodologies. How are you able to assess them and coordinate all those informations together?
Yeah, no, that's a very great question. So we started out with a big idea of how bacteria look and what we wanted to find out in really molecular details, which is like small scale compared to this bigger, just looking at something in a microscope. And we knew starting out that we couldn't just do this with the expertise in our lab alone. We needed to expand and involve people from other areas that use different equipment. And this has absolutely been essential to our work. It couldn't have just been done with one lab, it's been done with multiple labs.
What do you find most exciting about this work overall?
Yeah, so the most exciting thing, I think, is that bacteria are thought of to have this outer shell that is made up of lipid. So lipid is really quite malleable. It's quite almost like liquidy, like when you put detergent in something. So it's very fluid. And what we're saying, that contrary to the textbooks which say that it's exactly a lipid membrane, that in fact it's got these proteins in it. And these proteins are really important because they can allow something to pass into a bacterial cell. But in fact, these proteins do more than just transport things into cells. What we've shown is that they actually make up part of this outer layer of bacteria, and that they form this almond-like structure. So you've got lipids and proteins together, and there's like a hexagonal array around the whole shell. And this is quite a new idea, that you can have protein. It's a protein-lipid membrane that protects them and provides this from the outer layer.
Now, I know you've worked really hard and it was quite a long collaborative project. So what can you highlight the major challenge you face?
One of the things that was a real problem for us was trying to study molecular, so small scale molecule interactions in a live bacteria. So this was the thing. We weren't trying to artificially reproduce something that was already present in the natural world. We wanted to work on exactly what was happening in the world around us in these natural bacterial systems. So that was one of the big things, was making and developing a method for a natural bacteria and looking at this. But then also looking at two very different things. So a protein is very different to a lipid in terms of their properties and what they do. As you know, with your native mass spec, you're a specialist in these kinds of interactions. And so that was kind of the second problem, was actually working out how we could work with both of these and what techniques we could use to study both of the protein and lipid together. So prepping a sample that we could use and how we could apply this and study it.
For me, being part of this study is a very exciting experience. It's quite a long time, I will say, but it's quite intriguing when you can put together resources from different departments and expertise from different research groups to address a common theme. And secondly, I do think we learned a great deal by one lab preparing the sample, optimizing, another lab looking at the interaction of proteins with proteins and with lipid, and another lab looking at the same thing with another technique. I know we do mass spectrometry and others, so which is a great thing for me. Thanks, Melissa, for your time today. It was great chatting to you, and I found being part of this project a very enriching experience, and I look forward to us collaborating further. We have really a lot of resources in Kavli and in the department which you can use to take the study much more further. Thank you.